Hello, I'm Jan Cross-Zamirski. I'm a PhD candidate at the University of Cambridge, and I'm presenting our work, Self-Supervised Learning of Phenotypic Representations from cell images with weak labels. And this is a collaboration between the University of Cambridge and AstraZeneca. And here are the great scientists on screen who have contributed to this work. We are working in the field of image-based profiling, especially on this end, where we have images, fluorescent stain microscopy images, and we're looking to extract and learn meaningful profiles or phenotypic representations from these cells to perform downstream tasks. And we're using the BBC21 set, which has been used extensively with many models. And the key thing here is that there are 103 unique treatments, 38 compounds, and 12 distinct mechanisms of action. So here are the channels. And there have been many approaches taken on this data set, classical approaches such as cell profiler, transfer learning algorithms. But I want to talk about the weakly supervised learning, where the weak labels, I think treatment labels, were used as a classifier network. And then there was an embedding taken out of that, which was then used for downstream prediction. And that was quite successful. More recently, there have been unsupervised and self-supervised approaches, which are also very successful. And here's one example of those, where there's single cell crops actually with a contrastive learning framework. But we wanted to move away from single cell cropping and see if we could do this on image level, because there have been some studies that have done that with sort of multi-scale approaches. And we actually found, we sort of settled upon this algorithm, which is Dino from Caron. And we chose this for two properties that it has. The first property is, as you can see in the top left, very strong segmentation properties. And in the bottom left, it's also very good at clustering based on the features extracted from this network. And so we thought that it make it a good candidate for this sort of problem. And here's an example of it, sort of clustering ImageNet classes, obviously much more different data than what we're using. How does it work? Well, it takes sort of global, which is the red box, global crops from the image, and it takes local crops, which are the yellow boxes from the image. And it sort of has two networks which sees different sets of these crops and different augmentations. And then it sort of learns to maximize the agreement between these two networks. So it's sort of learning. It's sort of similar to a contrastive learning sort of approach. It's a knowledge distillation approach. And yeah, here's an example of us using it on the cell data. And we thought, well, how can we sort of adapt this to incorporate this weak label information that has been shown to actually enhance these features that we could learn? And we came up with weakly supervised Dino, or WS Dino. And we basically take the global crops from one image, and then we take the local crops from a different image of the same weak label class. And that could be same treatment, same compound, even same batch. Really, it's up to you which you choose. Yeah, we kind of wanted to retain the benefits of the powerful self-supervised learning approach, but also incorporate implicitly these labels. And we formulate this mathematically in the paper, but here it is. It's 2 large crops and 8 small crops. And we extract features from this network that we train. We actually fine tune. And the clustering properties are really good. And you can see here, these are the 103 treatments sort of aggregated and then plotted. And there are only a couple of them are misclassified. And these are known outliers in the data set. So yeah, we use sort of not same compound and not same compound batch matching, which are essentially one nearest neighbor algorithms to in the feature space using cosine distance, very accepted metrics in this data set. And we get the best performance using compound as the weak label. And we think the reason for this is that the compound sort of covers multiple batches, whereas the treatment doesn't. So maybe this has some sort of batch correction property, which could be really useful in other data sets. We also use the mechanism as a weak label, even though technically it's the strong label, just as a proof of concept, and that does it incredibly well. So compared to existing work, we perform state-of-the-art sort of level of results, and without using single-cell augmentation, which is generally the best performing results before us did. And we also can show that using attention maps that we're learning structurally meaningful features. This is a property of the vision transformer backbone of the network, which I didn't have time to talk about, sadly. Thanks for listening. Here's the great team. And if you want to know more about this work, scan the QR code and you'll see everything you need.